Hello, everybody, and welcome to the RNA Collaborative Seminar Series hosted today by the Bay Area RNA Club. We're going to wait another minute for everybody to join us. Hello, everybody again, and welcome to the RNA Collaborative Seminar Series hosted today by the Bay Area RNA Club. It's a sunny day here in Stanford. My name is Mary Krupkin, and I'm a postdoc at the Jody and Beta Puglisi Labs at Stanford. Please check out our upcoming talks at the RNA Collaborative Seminar Series, and keep your eyes and ears open for the BARC RNA conference coming up in December. We still don't know, but it might be in person, it might be virtual. So keep an eye out for that. Today, we will have two speakers with us today. We have Julia Salzman from Stanford University and Mark Ansel from UCSF. Let me remind you that you can use the Q&A feature to submit your questions today. Let me start with introducing our first speaker today. I am honored to present Julia Salzman, an associate professor at the Biomedical Data Science Department at Stanford University. She received many awards for her research, including the McCormick Gabillion and the Alfred P. Sloan Research Fellowships. She also received the Baxter Faculty Award, just to name a few. I'm excited to hear her talk today about the developmentally regulated RNA isoforms expression programs revealed by single-cell RNA sequencing. Julia, you have the stage. We can see your slides, but we can't hear you.
Hi. Okay, I think I unmuted myself, hopefully. That was key. I'm really excited to talk to you today. And I want to start by saying this is just a snapshot of our recent work. And I'd love any connections and contact if you want to contact me about any of the details, but due to the time, of course, I can't really go into great detail. This is the work of really a group of people in the group, talented, extremely talented postdocs, bioinformaticians, and graduate students depicted here. And I'm going to start by giving an overview of what motivates this work. RNA processing is really critical to gene function and gene regulation. And we, as molecular biologists, still, a lot of our discussion about the human genome and its products center around genes. But genes are really more complex than a one-to-one relationship between a gene locus and a protein or RNA variant. More than 95% of RNA of genes have alternative sequence variants, which we call isoforms. And from small numbers of examples of these isoforms that have been found by hand, in the sense that individual molecular biologists have really devoted huge amounts of resources and time to studying them, we know that isoforms of the same gene, having different isoforms of the same gene, is critical to development, proper development. It is having different expression of different isoforms is dysregulated and causal of many diseases, from neurodegenerative ones to cancer. And that normally isoforms of the same gene can produce proteins with different and even opposite functions. Okay, so because of the sheer complexity of this problem, the total number of genes, and the fact that sometimes dozens of isoforms are produced from a single gene, in order to get a picture of what the gene, how different isoforms function, we're really interested in using data science to do this, because it's almost impossible to do this using a one-by-one traditional molecular biology approach. And the first step in this program is one that is characterization. So what we're interested in, and I'll talk the rest of the time about today, is how we can use statistical bioinformatics to detect and characterize how individual cells and cell types regulate and co-regulate isoforms. And I'll tell you about two robust, simple statistical methods. Okay, before I do this, while many people are familiar with the way that alternative splicing of protein domains can affect function, I want to motivate you for the first part of what I'm going to talk about by giving an example of how the non-coding region of genes, for example, the 3' UTR, can have dramatic influences on protein function and cellular function. One of these examples is CD47. It's a cell surface marker, and it can protect against phagocytosis when it is exported to the plasma membrane. Okay, and so at a high level, you can take this toy example that there are two different variants of this gene. One has a short 3' UTR, and when it has this short 3' UTR, it stays localized to the endoplasmic reticulum. And this results in increased phagocytosis of such cells. On the other hand, there's a second longer isoform that when it is expressed, it goes to the plasma membrane and protects the cell from phagocytosis. Now, this is actually a simplified picture. In work that I don't even have time to tell you about today, we have found that there is highly regulated expression of more than just these two isoforms in human cell types. Okay, and then as I alluded to, the other kind of more commonly understood impacts of RNA isoform expression have to do with the produced protein variation. And one of the most exciting, interesting poster children for this is the enormous complexity of the splicing of the gene DSCAM in Drosophila, which is it's expressed in neurons, and basically serves as a fingerprint that allows each neuron to self-avoid in its dendritic growth. And if you disrupt the splicing of this gene, you get improper patterning of the brain. Okay, so despite this very fantastic and interesting example of DSCAM and CD47, the vast majority of RNA isoforms have unknown cell type specificity and function. And so what I'm going to tell you about are two robust, very, I would say robust in a statistical sense and also in a bioinformatics sense, two robust, efficient, and statistical approaches to finding regulated RNA expression in single cells. Okay, so I gave the example of CD47, two different UTRs. What would we seek to do from a single cell sequencing experiment? Ideally, we'd be able to identify that cell 1 and cell 2, if they indeed had two different variants expressed, cell 2 having the longer isoform, we'd be able to detect this statistically. And the goal here would be to kind of determine if cell types of cell type 1, for example, macrophages, might express a short isoform, whereas endothelial cells might express a longer isoform. And of course, we're interested in doing this automatically for any cell type. Just those are two examples. Okay, so how could we potentially do this? If there were reads, if cell type 1 indeed expressed the shorter isoform, what we would expect to see are a read distribution from a 10x experiment with a pileup close to the end of this shorter isoform, whereas in cell type 2, if it expressed the longer isoform, we'd expect a pileup near the longer end, the more distal end of the isoform. Okay, and intuitively, this pileup would be depicted in these kinds of histograms, and then we'd be able to identify these histograms, map the reads to the 3' end. And this is kind of the intuition that you probably are familiar with if you've thought about RNA-seq or single-cell RNA-seq. And this intuition is a good guiding intuition, but it actually misses really important signals that I'll tell you about. Okay, so, and it's the typical approach used in the field. Okay, so, sorry, I'm just looking at the chat and trying to move it. So what is missing in peak calling algorithms that are very commonplace in bioinformatics? For one, there could be very important regulation between two different UTRs in cell type 1 and cell type 2 that would manifest themselves as overlapping if this difference between the two ends is too close to each other. An example is here. And naive peak calling that would just basically quantify a human eye's intuition that is there a peak here or not would miss this kind of fine scale and potentially really important biological regulation. Okay, so what does the, right. And then the other point is that many algorithms approach this problem of determining whether two cell types have the same UTR use by pseudobulking single cells, aggregating all of the cells of macrophages together to get something that has more read depth. And of course, this loses power because these algorithms cannot distinguish whether cell types are, each single cell is expressing two different isoforms, or like maybe with 50-50 mixture, or half of the cells are expressing one isoform and the other half are expressing the other. Okay, so our approach is called the ReadCS, and it has some important differences between every published method and approach to this. And what it does is it takes us, it is a bona fide single cell resolved measure of how distal a UTR is. And basically it tiles, and it's also annotation free. What it does is it tiles the genome into windows. Here I'm showing in blue reads from cell A, and in orange reads from cell B. It takes every single read and maps it to the genome. And you can think of what it does. If you're interested in the statistics, you can read the paper, or I'm happy to explain it in Q&A. It maps, it basically aggregates these reads in a cell type in a statistical way, and compares whether and gives a cell score that basically summarizes how distal the read distribution is within this window. And what this enables us to do is both test for differences in UTR use between cell types, and also detect developmentally regulated UTR expression. Okay, wow, I'm at the 15-minute mark. Okay, so I'll try to speed up. ReadCS has been able to identify, so what we can do with this is basically correlate the ReadCS score at a single cell level to pseudotime to determine whether there are genes that have regulated changes in their UTRs over developmental time. And we found a huge number of examples of this. Giving an example here, many of these regulated events are conserved across evolution from human to mouse. This is a GNZ FAN6, and you can see there's a strong shift in both human and mouse from early, like a longer isoform early in development to a shorter one later. Okay. Another thing that this approach allows us to do is really get a fine scale picture of developmental regulation that is missed by pseudobulk. So here what we're doing is we're taking a single developmental time point that every pseudobulk type algorithm would aggregate together, and showing that genes like OAZ1 and MED1, which have important functions in chromatin organization and mitosis, actually can have detected regulated UTR use. And this allows us also, I should say, the bona fide single cell nature of this allows us to do de novo clustering of cells, including subclustering of cell types. Again, this is work in preparation I don't have time to talk about. I'm going to give a very quick 3-minute overview of another approach that is now published called the splicing score, that is very similar in spirit to what I just described for the READ-TS, except that it measures how different splicing choices are in different cell types. Again, here we have, this is a bona fide single cell resolved measure of per gene per cell. And it allows us to do the identical kinds of analyses to detect developmental regulation, cell type specificity of splicing, and also subclustering. I'm going to gloss over that and just give the bottom line for one of the, I think, most interesting fallouts of the application of this, which is to nearly 100,000 human cells and many tissues as part of what's called the Tabula Sapiens project. And what we found, which was very surprising to us, is that essential housekeeping genes have tightly regulated splicing at cell type resolution. And two of these genes are, one of these genes is MYEL6, which is a myosin light chain gene. It's ubiquitously expressed. And you can see from this picture that it has low inclusion of one exon in stromal cells, for example, and high inclusion in epithelial cells. We've validated this. Another super interesting example is a gene, tightly regulated splicing of the ribosomal protein gene RPS24, which is an essential ribosomal protein gene. Again, I'm not going to have time to talk about it, but there's a very tightly regulated microexon. And then finally, I'll close by saying that all of these measures, while we've developed them for single cell sequencing, they can readily be applied to spatial genomics like Visium. And we have now been able to show that splicing of some of these housekeeping genes like MYEL6 is spatially regulated. You can see that in this kind of, I don't know, pseudo-colored histology, pseudo-colored on the basis of its splicing value. You could think of it as exon inclusion rates in MYEL6 here in the brain, here in the kidney. And there's a very stark, regulation here. Gene expression is not correlated with these values. And yes, so in conclusion, we are excited that I think this opens the door to more wide use of these algorithms to study RNA processing and regulation. And I thank people in the beginning. I'll thank them again, and as well as funding, and take questions for a minute if I can. Thank you.
Thank you so much, Julia. This was a wonderful talk. It's so fascinating to hear about RNA splicing. I work on HIV viral RNA, and I would like to ask you, can we apply these approaches to study the alternative splicing and isoforms of viral RNAs inside the human cells?
Yeah, thank you for asking that. Definitely, because all of this is basic, it's all annotation free. So all you need is a reference genome, and actually an unpublished work. So we're very near each other. We should talk. We have a way of doing this without any annotation at all, including without even a genome annotation. But this work is based on only a genome annotation.
Oh, that's really good to hear. Thanks. I have a question, and I want to remind everybody to use the Q&A feature. I have a question from Benny Mosquire. He's asking, how can you use this awesome tool? Is there an additional step, or is this the RNA-seq pipeline that anyone can implement?
Oh yeah, we have it actually, thanks especially to Caitlin here. It is all implemented in Nextflow, which is an incredibly easy to use pipeline. So we have both of these methods are on GitHub or even some other portals. If you want to, please try to look at our bioRxiv and Nature Methods papers. And then if you can't find it, just contact me.
Sounds good.
just.
Well, thank you again, Julia. And if anybody has more questions, well, maybe we'll have more time at the end. So I would love to move to our second speaker today. Let me introduce Mark Ansel, a professor of microbiology and immunology, and an investigator of the Sandler Asthma Basic Research Center at UCSF. He's a co-founder of the UCSF's ImmunoX initiative, and also a co-founder of UCSF Propel program, a program for post-baccalaureate research opportunity to promote equity in learning. I'm very excited to hear his talk today about dissecting post-transcriptional circuits in immune responses. Mark, we can see your slides and you have the stage.
All right, thank you so much. And also thanks for your forbearance as I ran around trying to find a place to do this. My internet amazingly shut down in my whole lab. So if there's background noise, you might want to let me know. The best place I could find is not the quietest. So let me know if things don't sound good, just barge in. So it's great to speak to an audience of RNA biologists. We are RNA immunologists in my lab, and it's a fun time to study RNA in immunology with the success of the RNA vaccines. We're really interested in understanding the physiological regulation of immune responses by RNA binding proteins, by small RNAs, long non-coding RNAs. And we like to take a cis-regulatory approach to understand how these things work. So I'll talk today about biochemical and functional mapping of the regulatory transcriptome. I won't talk today about microRNAs or about our other immunological studies in airway and allergic inflammation. But I will tell you a little bit of immunology in the end about a particular cis-regulatory element that we think is really important for lymphocyte homing and ultimately for the susceptibility to autoimmune diseases. The work I'm describing today is almost entirely the work of Didi Zhu, and I'll mention other people who've made big contributions as we come to it. She's a senior graduate student in the lab who's really led these studies forward over the last several years. So I don't have to describe this too much for this group, but of course, post-transcriptional regulation is extremely important to control the duration and the degree of expression of genes that get transcribed in different cell types. RBPs can do a lot of different things. They're the transit factors that bind to RNAs and can mediate changes in their ultimately amount of protein that they make through changes in splicing, as we just heard, but also a very prominent role in changing the rate of degradation of the mRNA, can stabilize as well. And it can also move RNAs to a different part of the cell for translation there or to prevent translation. And these RBP interactions are guided by cis-regulatory elements, either linear or structural elements in the RNA that could be recognized by these proteins. RBPs are certainly important in the immune system. And there are several cases out there. This happens to be my favorite one. So the Roken protein, that's named for San Roque, the patron saint of plague victims and dogs and all kinds of other interesting things. Plague victims, people who get the plague, get really big lymph nodes. That's the buba. And Roken mice, which is a mutation that was created in Chris Goodnow's lab a long time ago and studied by Carola Vinuesa, get really big spleen and lymph nodes spontaneously. And it turns out to be because of interruption of the activity of two proteins called Roken1 and Roken2 that control immune responses and prevent them from running away and causing this lymphoproliferation. And these mice end up with a lupus-like syndrome because of this single mutation. And there are lots of other ones that are important. And of course, all of the work that we've done on microRNAs is really just a special case of an RBP, Argonaut proteins that are guided by small RNAs. Individual cis-regulatory elements can be really important too. And this is not an example from immunology. This is an example from agriculture, where people have figured out that these prized texel rams that get really big muscles do so, at least in part, because of a single nucleotide transition in the 3' UTR of the myostatin gene that creates target sites for microRNAs that are really highly expressed in muscle. And so they grow muscle out of control. And there are also really good examples in immunology, and the Rudensky lab, the Turner lab, Papadopoulou, and Nussensweig labs have all done experiments in mice where they change individual cis-regulatory elements and see physiological effects on immune responses, leukemiogenesis, immune cell development, and other things, but always in a context and cell type dependent manner. So there's a lot to learn, and we know so much less about cis-regulatory control of post-transcriptional gene regulation than we do about transcriptional gene regulation. If you just showed you a generic piece of the genome and said, where are the cis-regulatory elements, you'd say, well, you know, where are the regions of accessibility? Give me the ATEC-seq tracks and the histone modifications and DNA methylation. We've got so much data out there from so many cell types from the ENCODE project and others. And that's really paved the way for systematic analyses of enhancer function. And it's helped us to understand that a lot of the variation in the genome that has effects on physiology and health and disease end up landing in these regulatory elements that affect transcription. But if I tell you that this is a generic mRNA, you'd say, well, it's probably mostly in the untranslated regions. We could look at some things, and there's been a movement since I first made this slide. There's been a movement towards doing a lot more of this. There's been the ENCORE of the ENCODE project for RNA, and we're starting to build up biochemical information in a few different cellular contexts for many individual proteins, RBPs, and where they bind. Some things about RNA structure, some things about RNA modification even. But we've lacked the sort of ATAC kind of experiment to say, what are all of the interesting bits of this piece of mRNA from a regulatory standpoint? A few groups have worked on finding all of the RNA-bound regions of mRNAs, and it was done in yeast first, and then a couple of different groups have developed good techniques that work in mammalian cells. Most recently, I didn't list them here, I'm sorry, OOPS and scRNA-seq are phase separation methods that pull down proteins and RNAs together and help you to map what RNAs are bound. We developed a technique called GClip based on this Freeberg et al. Paper that was first done in yeast, where we basically do a clip experiment to find out where a particular protein binds the mRNA. But then instead of using an antibody as an affinity reagent to pull down cross-linked protein with its RNA-bound regions, we just capture all of the adenylated RNAs and biotinylate all the proteins that are attached, specifically attached. And then we can elute that, chew up all of the extra RNA, like you always do with clip experiments, and pull down all the proteins using these biotin tags. And so you get more material, you get a lot more binding sites. This is real data over here. And these ones down below are two examples where you can see sometimes an mRNA is really highly decorated with proteins. So when you sequence the RNA, you get peak after peak after peak. And this top one, IER3, turns out to be one that's a known ROCN target. And that little stem loop there is the ROCN binding motif sitting in this big peak. NR4A1 is a different kind of example where you can see that there's really discrete regions of binding. And these little red boxes indicate canonical AU-rich element binding sites that are known to be very important for the rapid turnover of this mRNA and many others that are important and have pulsatile expression in lymphocytes. So we've done a lot to validate that GClip works. It finds real binding sites. We believe like taking the average of all possible clip experiments and just getting all of the data at once. If you look at where the reads come from in a GClip experiment, you see it mostly in 3' UTRs. Link RNAs are also heavily covered, and also quite a bit of protein binding in exons and 5' UTRs as well. So I'm skipping over all of the validation, but we benchmarked this against scRNA-seq and OOPS and other methods, and looked especially to see if it recapitulates what's there in the ENCORE data set provided by GeneYeo, the ever-growing ENCORE data set with a bunch of clip experiments. And it looks like it creates transcriptome-wide maps of RBP occupancy. So we do a lot of interesting things with that. I can't talk about all of them today. We can start to look at differences in different cell contexts and different cell types. We can assign biochemically difficult transacting factors where no one has a good antibody to their cis-regulatory elements using this technique and some genetics, we can start to integrate this information with all those other things I mentioned before and provide resources that people can use to follow their favorite genes. And of course, we can look at our favorite genes and start to predict and test whether some of these putative cis-regulatory elements that bind proteins in messages, sequences in messages that bind proteins are indeed cis-regulatory elements that affect the expression of those genes and their physiological functions. So we did that first on a broad scale using massively parallel reporter assays, and more recently, we've started to prioritize candidate genes using things like genetic data from GWAS studies, cancer genomes, and this kind of thing. So this work is published some time ago now. So the massively parallel reporter assay, where we took all of these GCLIP peaks from T cells and stuck them into a reporter assay and put those reporters back into T cells, uncovered some really interesting evolutionary patterns in what the content of these regulatory regions and their activity on destabilization of mRNAs, and particularly things that were GC rich and folded a lot were much more likely to be destabilizing than those that have a U-rich sequence. And that agreed with work from Alex Shear's lab that came out around the same time, work in zebrafish embryos using a totally different system. And so I think we're really looking at regulatory elements here. So for the rest of this talk, I'm just going to focus on one of our favorite hits, and it's one of the ones that we got interested in because of the work that Didi did to intersect human genetics with our GCLIP results. So going back to transcription and enhancers and all of this great mapping that had been done, Alex Marson and Kyle Farr several years ago now used those maps from a bunch of different kinds of immune cells of the regions in DNA that are accessible and intersected that with SNPs that are associated with disease. And they didn't just use all the SNPs. They used a probability algorithm to identify the most likely causal SNPs. So we narrowed it down to things that by the PICS algorithm were at least 5% likely, at least had a probability at least 5% to be the SNP that actually leads to susceptibility to whatever disease they were looking at. And what they found is that these SNPs were greatly overrepresented in things that were mapped as enhancers using ATAC and other methods like that, chromatin histone remodeling. So, we asked, can we do the same kind of thing with GCLIP instead of ATAC? And so there's a smaller number that map to 3' UTRs, but there's still quite a few probable causal SNPs that map to 3' UTRs. We updated this because there's been a lot of GWAS completed, actually as much since this paper came out as before the paper came out. And so now we have an updated guide for probable causal SNPs for immune diseases that was put together by Kim Taylor in collaboration with Kyle and Alex and Lindsay Criswell. And then we intersected that with our GCLIP peaks that are present in T cells to ask if things that are regulatory in T cells might be responsible for disease susceptibility. And indeed, we found a bunch of things. I don't have time to show you all of them. We've validated that several of them have regulatory activity. And I'd really like to focus in on our favorite one, region in the CD69 gene. So we picked CD69 for a couple of reasons. First of all, it contains several RBP-occupied sites. So this is a GCLIP track for CD69 in these Jurkat T cells. You can see there's several discrete high binding peaks. Secondly, the CD69 mRNA has a really short half-life. It's upregulated really quickly when lymphocytes get activated, and then just as quickly, it goes away again. And it's a very regulated protein from transcriptional level through post-translational effects on its ability to stay on the cell surface. And most importantly, there were two disease-associated probable causal SNPs in the 3' UTR. You can see them with the arrows here. One is associated with type 1 diabetes. The other one is associated with multiple sclerosis and has a 79% probability of being the causal SNP for susceptibility to MS. with an odds ratio of 1.1. And they both hit regions of RNA binding. They're not the highest ones, but they're consistently and significantly enriched peaks in these GCLIP tracks. So just a word about CD69. It has a really important, highly regulated role in T cells. The work of Jason Sister, Matt Lubian, and others showed how CD69 promotes cell retention in lymphoid organs and in peripheral tissues through interactions between CD69 and sphingosine phosphate receptor S1PR1. So CD69 and S1PR1 localize together and interact with one another. And basically, whichever one is expressed more wins and ends up on the cell surface. So you have lots of CD69 in the cell, and S1PR1 isn't on the cell surface, and cells can't sense the S1P gradients that lead them back into the bloodstream to go circulate around. And this has been taken advantage of with pharmaceutical companies. The drug fingolimod, it's also called FTY-720, causes S1PR1 downregulation and loss of its activity. And that traps cells in lymph nodes and in various organs, and it disallows them to be involved in causing inflammation in distal sites. And thereby it's a really potent immunosuppressant, and it works really well for MS patients. In fact, it works so well that it has some safety problems that it can reactivate viral, latent viruses in the central nervous system, because it's so important to control this system to maintain proper immunity in the CNS. So that made CD69 and its genetic connection really interesting to us. And we also knew that there must be a lot of regulation of this transcript, since it doesn't hang around very long. So our goal was to figure out which of these cis regulatory regions are important and evaluate their effect on T cell biology. So DD first took a GCLIP directed approach to CRISPR-mediated editing of the 3' UTR of CD69 and JurCat T cells. And you can see with these little arrows where she developed guides that had good high efficiency deletion, and she cut out all 4 of these regions of the CD69 3' UTR. And this is just the 3' UTR you're looking at here. And she made clones of JurCat so you could see the full effect in cells that were completely lacking these regions that were sequenced, so we knew what the deletions were. And the most important result is shown here. This is an mRNA decay experiment using actinomycin D. And you can see that the half-life of wild type CD69 and JurCats is super short. I think I had 30 minutes on one of those slides. It's actually more like 15 to 20 minutes, maybe 30 minutes in some experiments. So it's a very short-lived mRNA and decays very quickly if you can't keep transcribing it. If you delete the whole 3' UTR, so use these most outermost guides and just maintain the very end and the poly A, then the transcript half-life gets much longer, more than an hour in JurCat T cells. And interestingly, if you take out individual regions here, you can see that this one, the B region, has almost all of the activity. C and D have some activity. B has a really strong activity on CD69 half-life. And you can see this reflected in the protein levels. The gray is the amount of protein on a wild type JurCat T cell after it's activated. The 3' UTR knockout increases a lot. So we know there's a lot of regulatory activity in there. And a good bit of it can be attributed to this B region. And you can see the green here is shifted to the right. So more at CD69 than any of the others. That's flow cytometry data. And there it is in a bar graph with some replicates. You can see that B seems to be the most interesting region to follow. And it turns out that B is also the region that contains those SNPs. So we continued to work on B, and so far we've pretty much ignored these other regions, C and D. Importantly, a lot of this biology is shared between mouse and human. It's pretty deeply conserved, it appears. Didi did a similar experiment using primary mouse T cells. Here she's using Greek letters instead of Arabic alphabet. And so, you know, Arabic alphabet instead of our alphabet. And And she has very similar results. Maybe beta doesn't have as much of an effect as taking out the whole 3' UTR, but it clearly is the most powerful regulator of CD69 protein expression in mice as it is in humans. And if you overlap the human and the mouse 3' UTRs and look at the G-CLIP peaks and also at sequence conservation, you'll see that B and beta overlap, and they overlap basically in 2 protein binding regions that are also highly conserved. And so this is the phylo-P score down below, and these big peaks indicate regions of high conservation that associate with these binding peaks. Their relative intensities are different, but there's great overlap between the binding activity in both species. So we went ahead and made CRISPR knockout mice that targeted just this region beta in mice, or like we did in jerkats, the whole 3' UTR without disrupting the polyadenylation signal sequence. And if you take those cells and put them, the T cells that result from those mice, and put them in vitro and activate them with immune stimuli that will get them to upregulate CD69, you can see that first in black, the wild type ones activate CD69. They upregulate it about 5-fold at the mRNA level. The 3' UTR knockouts upregulate more like 20-fold. And so even within those 4 hours, there's already been a big effect on the transcript levels just by changing, by taking away the 3' UTR regulation. And it's a longer sustained expression that stays on throughout the time course. The beta knockouts had a phenotype. It was just not as strong. So this is a part of what's going on in the 3' UTR, an important part that's significant compared to wild type. We also asked, could this have something to do with the promoter activity? Is it different when the cells are no longer being stimulated? Is it somehow regulated by stimulation? It looks like these elements are important at rest as the cells decay away all of their CD69 transcript, as it is when you're activating them. So it's a regulatory element that continuously reduces the stability of CD69 mRNA and activated and resting T cells. You can do the same thing, look at protein. It's the same kind of story. You can see that there's more expression, especially in the 3' UTR knockout. And especially at rest, we're able to see significant effects in the beta knockout until it reaches the bottom. And basically what happens out here at these later time points is that S1PR1 catches up and can to extinguish the CD69 signal on the surface of the cell. So that's to remind you that we're interested, or to remind me that we're interested not only in whether this element actually regulates CD69, but whether that regulation actually matters for the physiology of the T cell. And so we started to look at some of the things that CD69 does. And it's known from the literature that increased CD69 expression traps developing T cells in the thymus. That's the first place where one should look. So T cells develop in the thymus, which lays over the heart. And when they're fully mature, they gain the ability to respond to S1P gradients and get out of the thymus, move into the peripheral circulation where they go through lymph nodes and spleen. Now the expression of CD69 is increased in these mice that have either complete loss of the CD69 3' UTR or just this one element. And so we wondered whether that would make a difference for thymic egress. So yeah, if you simply overexpress CD69, this is in the literature, just straight up overexpress it with a cDNA construct, or if you knock out S1PR1, you get T cells trapped in the thymus and fewer of them get out into those peripheral organs. And so then we asked, how does this affect T cell egress? Is it part of the way that T cells get rid of their CD69 so that they can leave and go get involved in immune responses? So Didi did this in the intact mice, but for the experiments I'll show you.
Mark,
yes.
3 more minutes.
Yep, we're almost there. So she used an immunological trick to in the experiment I'm going to show you, where she mixed bone marrow from wild type mice and from mice that have these different deletions in their CD69 3' UTR, and then asked how those cells did together in the same mouse. So it takes away a lot of the variability in these experiments. And so you can do that with a mixed bone marrow transplant. And so if you look at the cells in these experiments, the wild type cells look like this. You've got CD8 T cells and CD4 T cells in the thymus. These are the two types of T cells. And you can already see that there's reductions in some of these cells in the peripheral lymphoid organs of the 3' UTR knockout and the beta knockout. Particularly the CD8 and CD4 T cells are reduced, CD8's even more than CD4s in the spleen. And the same is true when you go and look in the lymph nodes. And so instead of them piling up in the lymph nodes, we're seeing them missing. And so we imagine that might be because they're getting trapped in the thymus. If you look at the thymus, indeed, there's more cells in the 3' UTR knockout that are piling up in these CD4 and CD8 single positive compartments. And so it's the reciprocal effect of the last one. You see lots more cells here. And I'll point out that there wasn't a big effect of the beta knockout here. We can try to get excited about some trends, but actually we're more excited that the beta knockouts don't have a huge defect in the thymus, even though we certainly see changes in expression. So in the beta knockouts, this blue line, you can see that the cells expressing CD69 are brighter. But in the 3' UTR knockout, not only are they much brighter, but the cells that should be keeping it off are now turning it on. And so we think that this is why they're getting trapped in the thymus. And it also means that in these mice, we can start to ask about immune response in the periphery. So for example, we're interested now in doing diabetes and multiple sclerosis models to ask whether this region and the duration and intensity of CD69 expression because of post-transcriptional regulation affects the occurrence of diabetes and MS as predicted by the human genetic data. So that's all the data I want to share today. We've generated a map of RBP binding sites in the T cell transcriptome and used it to identify regions in genes in 3' UTRs that appear to be important for regulation of the corresponding mRNAs. We studied CD69 and we found that it's really important to have post-transcriptional regulation as well as transcriptional and post-translational regulation of CD69 to control its levels and how long and how intensely it's expressed so that cells can move around as they're supposed to during their development and in immune responses. So we're still working to identify the RBPs that bind to this region. We've taken a proteomics approach with help from Monica Walkers. And we have some great candidates that we're following up on. And as I said, we're really interested in moving this into disease models to understand how this region identified by human genetics affects T cell function in immune disease. So again, I want to acknowledge Didi, also Adam Litterman, a postdoc who started the GCLIP project, and Sean Second, who's helped us with a lot of the bioinformatic analysis of these data. I have a great lab of people. This is a slightly older picture from our last lab retreat. We're going back for our first lab retreat in a couple of years to Monterey in the next week. And I'm really happy to share this with all of you. I hope I can answer some of your questions. Thanks for your attention.
Thank you for a fantastic talk, Mark. And I can't wait to hear more about RNA binding proteins in diabetes and MS, because of course it's so important to learn more about it. I want to mention we do have a few more questions for Julia, but we'll start with questions for Mark first. So Julia asks, when you perform deletions, do you verify that the targeted genomic deletions actually cause the UTR changes you predict? It could be deleting an actual result in a longer isoform due to an impact A, could have on the regulation of transcript cleavage or poly A length. Mark, you're muted.
Hi.
Hey.
I'm sorry, I thought you were asking a question for...
Oh, so the first question for you was from Julia.
Oh, I see.
I can repeat the question?
Yes, please, because I don't see it in the Q&A.
Okay, let me, you can see it in our chat, I think. Your question is, when you perform deletions.
Okay, I see it. Does it change localization or does it cause changes in the translation or no? You mean the one from Julie. I'm sorry, I thought you meant you were going to ask her a question from her talk. So do we verify that we're getting these deletions are the ones that we wanted? And the answer is yes. We can't, when we do that in the primary T cells, we can't ensure that every one is the right deletion. But what we can do is look in bulk at the PCR products and then sequence what we get and see if we mostly get the right kind of deletion. With the Jurkat experiments, and of course, with the transgenic mice, we can sequence and know exactly what we're working with. So in those cases, we do that and make sure that we have precise knowledge of what we've deleted.
Maureen Akini asks, are there CD69 splicing isoforms, and how does shortening or messing with the 3' UTR affects this, the splicing isoforms of CD69? Can you reliably trace the specific 3' UTR knockout expression to protein expression, such as excluding compensatory effects?
Well, first of all, I'm not aware of CD69 splicing isoforms. There's not a lot of evidence that there would be any. I can't rule out that there couldn't be some in some cases, but it's not a gene that's known to be regulated that way. So it's a good reason to choose it, right? And splicing is very interesting, but I'd rather not have the complication. Can you reliably trace to protein expression? By compensatory effects, I assume you mean that there would be some kind of feedback if you took out the 3' UTR that would somehow affect transcription. And we've tried to rule that out by looking at mRNA half-life in particular, and that correlates well with the protein. There's no question that CD69 is being regulated at the transcriptional, hyper-regulated at the transcriptional and post-translational level. If I had time, I'd show you some things about how there's a threshold effect. When the protein reaches a critical point, then it gets trapped inside the cell and you see S1PR1 instead. And we see that happening as the protein level drops in physiological context where S1PR1 is expressed. So I think we're mapping this correctly to protein expression through changes in mRNA degradation. But I can't exclude that there couldn't be some other things going on with localization of the mRNA, or I guess even some kind of transcriptional elements hidden in the 3' UTR.
I see. There's a similar question coming up from Ashley Byron. She asks, does mutating the CD69 3' UTR region changes the localization of CD69 inside the cell? Do you see it cause any changes in the translation levels?
Yeah, we see it lost from the surface, but when the two proteins interact, it also leads to their degradation. So it's not like we see it pile up inside the cell, but we certainly do see that it gets lost from the surface of the cell. We did not measure translation per se. This does seem to have a really powerful effect on the transcript, on transcript degradation, but my bias is that probably involves translational repression, maybe even translational repression that leads to mRNA degradation. So that's a really good question. I don't have specific evidence to say for sure that a translational block is what's happening here.
We've got a lot more questions coming up. Let's try another question for Julia. Julia, can you hear me?
Yes.
Ah, wonderful. Mariela Cortez Lopez asked you, can this method be scaled for long-read single-cell RNA sequencing?
Yes, for example, for SMART-C2, both of these methods can be, and especially the splice actually has more powerful versions that I didn't have a chance to talk about that work better with SMART-C2. And yeah. Interestingly, I would just note, I do think CD69 actually does have splice isoforms, and I think it has, in a fun twist of events, I think it jogged my memory that I think we found it as one of the regulated examples in the analysis that we've done genome-wide. So that's a fun note.
We should share data and see that.
Yeah, definitely. Oh, yeah, we're happy to.
Well, there's a lot more questions in the Q&As. And I encourage you all to approach Julia and Mark, send them an email. Those are fascinating questions, but we're running out of time. We heard a lot today about RNA processing post-transcriptionally, and I want to thank Julia and Mark for sharing your research, your methods, and your computational tools with us today. I want to thank everybody who came in to listen and learn, and I want to encourage you to join us in upcoming seminars. A round of applause to our speakers. Oh, and today's recording will be available online soon. So you can check the YouTube channel of the RNA Collaborative Series seminar. Say hello again.